Great. It's also a real pleasure to have a chance to talk to the folks at Dana-Farber today. And I'm actually going to describe two different approaches that we're using for delivery systems using nanoparticles. And in fact, Bob gave a wonderful talk about the power of nanoparticles. So I'll describe some of the things that we're doing to look at different modifications of these systems. We actually have a technique in our lab called layer-by-layer assembly, which allows us to assemble oppositely charged species in alternation. And we can actually generate very nice systems that contain high concentrations of drug in these layers. Well, we've been doing this with nanoparticles as well as on macroscopic surfaces. And the nanoparticles can begin with a core which can contain a drug of its own, for example, paclitaxel. It can be a core which is a quantum dot or is some other signaling molecule. We can build on top of it layers which contain, right now we're working on siRNA and some molecular inhibitors. And finally, we can have an outer shell, and we can use that outer shell for the stealth property that we can get from something like PEG or from a natural polymer such as polysaccharide. Now this actually shows some in vitro circulation and biodistribution and stability of some of the nanoparticles that we have. And we have some systems now which exhibit very nice biodistribution in the mouse. We've actually looked at different polysaccharides as the outer coating, and we found that hyaluronic acid among some of the systems we've looked at actually give us very little accumulation in the liver. I should look over here too. Here we see the comparison between our nanoparticle with hyaluronic acid, some of our other nanoparticle systems, and the quantum dot itself. So we're actually interested in the fact that we may be able to lower some of the liver uptake, which is a natural side effect of using nanoparticles. We tend to get a greater amount of accumulation in the filtration organs by using these native extracellular matrix polysaccharides, which tend to give us a greater degree of self-recognition and protection from phagocytes. We also get very nice EPR effect, that enhanced permeation and retention effect that allows nanoparticles to accumulate specifically in solid tumors. And this shows you some of that accumulation over a period of about 24 hours. We also see that this, again, hyaluronic acid gives us a very nice long distribution time in the bloodstream. So we've been looking at different ways of manipulating these systems. One of them is to actually use, rather than molecular targeting, the tumor microenvironment as a way to accumulate the nanoparticle into the tumor. In this case, rather than using a molecular targeting ligand, which is something that we can do with these nanoparticle systems, we can also generate or design a system which has a dynamic stealth layer. In this case, we can have, for example, a top layer that is positively charged, poly-L-lysine in this case. And we can then top it off with a polymer chain, which gives us a stealth property. In this example, we actually used polyethylene glycol. We conjugated it to aminobiotin, and we can actually use avidin to conjugate that to the polylysine with another aminobiotin linker. So with these linkers, we actually have a very strong interaction, but it's pH sensitive, unlike the typical biotin-avidin interaction. When this circulates in the bloodstream at pH 7.4, it's relatively stable, and it gives us the stealth property that we want. But when we get to a tumor that is slightly hypoxic, even a small drop in pH causes this to shed off, and we unveil the positive charge. That positive charge causes non-specific interaction with the cell membrane that causes a clathrin-mediated endocytosis to take place and uptake of the nanoparticle. So you can see, for example, after 24 hours, a tumor on the mouse flank. This is the non-pH sensitive linker, and this is our pH sensitive linker, and we have a good deal of accumulation in these systems. This shows time frames. You can see the EPR takes place in both the pH sensitive and the non-pH sensitive linker, but we get higher accumulation over much longer time frames with the pH sensitive one because we're getting actual cell uptake of the nanoparticle rather than simple circulation in the intratumoral space. And this is just a bit of a cartoon showing that shedding of the layer in the hypoxic region. We've also been looking at ways in which we can do combinations of drugs, and some of the work we're doing with my colleague Michael Yaffe at the Koch, for example, we're looking at combination drugs that can address breast cancer by timing or staging the release of drugs. Now, on the other hand, we're also very interested in being able to replace the kinds of drug carriers that are traditionally used for nucleic acids. And there's been a good deal of work and beautiful work done with both polycations and cationic lipids for bundling these negatively charged siRNA fragments. And this has allowed us to move nanomedicine a little bit further along and to move RNA a little bit further along. But we still have some limitation in terms of our targets. We can address liver and a few other organs fairly well with RNA now. But there are concerns about toxicity when we begin to develop carriers to address cancer and actually have some sort of systemic delivery mechanism. The reason for this is that typically the amount of cationic lipid or the amount of charged polymer that you need to encapsulate a significant amount of siRNA is large, and it tends to be toxic. So when we get to the therapeutic window that we work in, we end up with the need to have a large amount of siRNA, relatively speaking, for a therapeutic effect, but a relatively small amount of carrier, and instead we end up with the opposite. 40 to 1, sometimes as high as 40 to 1 carrier to RNA. We'd like to find a way to replace these kinds of traditional carriers. And to do this, we actually looked at how we could actually design an RNA molecule so that it was extremely long, could actually pack very densely, and could then be delivered in a very compact package. In other words, we're trying to turn things upside down and have a package which is primarily RNA with a small amount of positively charged polymer that can be used in the end to help with transfection, as opposed to a package which is primarily cationic species. So typically, we would make our RNA with a traditional RNA polymerase, which would give us these short 21 to 22 base pair species. However, in our lab, we've actually been using a rolling circle transcription method, which uses a circular strip of DNA that has been coded for the sense and the antisense RNA. And in between, we've placed a short link that is cleaved by dicer, which is the enzyme that exists in these cells that naturally helps to cleave RNA. We then use T7 polymerase, which allows us to continually knit this RNA strand out to very large copies. Typically, we believe we're getting close to 500,000 copies of the RNAi that we're interested in these systems. And this is just a little bit of a cartoon of what's going on here. These actually form macromolecular species that have a superstructure that's very porous. They tend to break off into these microspheres. Here you can see an RNAi sponge particle. We call these sponge particles because they're about 2 microns, but they have the porosity that's very similar to that of a sponge. And you can see some of the features. These fold-like features we found actually correlate to the length of the RNA strand. So we believe we have these hairpin RNAi structures separated by short dicer linkages. And you can see when we actually introduce to this very large, dense molecule a very small amount of polycation, because it's porous and negatively charged, it shrinks down into a nanoparticle, about 200 nanometers in size. And we can then deliver these systems, and what we see in vitro is that we can get very nice effective transfection. And from my angle, it's a little bit hard to point this out, but you can see comparisons here between siRNA that is introduced alone at different quantities or molar concentrations. siRNA with lipofectamine in vitro is sort of the gold standard. Would never use it in anyone's body, but it works well for cells. And then we have RNAi microsponge without the polycation wrap and then with it. And what we're seeing is that we can get knockdown very similar to the lipofectamine. But what's really interesting here is the concentrations that we're using. If you look here, we have 100 nanomolar for the lipofectamine, and we're looking at essentially 1 picomolar for our RNA microsponge. So we're looking at a 3 order of magnitude lower dose, essentially, and getting the same amount of RNA in. We have a much smaller amount of carrier needed to get our target. And we have found that this does work in vivo. This is just a simple luciferase knockdown, which shows that over a period of 3 days, this is actually with intratumoral injection directly of these positively charged species, we can see knockdown of the luciferase gene. Right now, we're actually moving forward to look at some of the mechanisms of breakdown of the RNA nanoparticle. We think we can make it even more efficient. We can get even higher knockdown for this very highly potent system. We believe that we can actually improve the wrap or the carrier that we're using, the polycation. And we're now looking at a range of very specific genes that we think are important for ovarian cancer, and we're very interested in exploring some of the other systems that we can work on with this approach. So I'll stop here, and maybe there's time for questions. I'm not sure.